So, hello everyone. We are once again in the What We Are Reading YouTube channel of the South American Mycorrhizal Research Network. I'm Cesar Marin, and today we have with us Professor Matthias Rillig from the Free University of Berlin in Germany. Matthias is very well known in our field for many years now, and he has his own YouTube channels. Can you tell us which are these YouTube channels?
Yeah, it's the Life in Academia YouTube channel where I've got now maybe about 200 videos about what it's like to be in academia. I also have a new YouTube channel that is called Life in the Soil, but it's just kind of developing. There's only a few videos on there now.
Excellent. I will put down these YouTube channels in the description. Um, well, as you may know, and over a couple of years ago, we did the first interview here in the same YouTube channel with Justin Cars and Melanie Jones and Jason Hoexima about common mycorrhizal networks. Has been a huge topic in our field, especially since that paper and several others that have appeared. And recently I even saw a kind of a response article by Susan Zimmer. So is the field is moving along with kind of different views on these networks. And Matthias Group has been producing as well. How many papers already since 2023 about common mycorrhizal networks?
Um, I think there was the opinion paper about the redefinition, and now also a systematic mapping paper that looks at the literature that's been published on this topic so far, and I think the others are still in review.
Okay, great. Yes, I think that I saw two or three articles, and today specifically we will focus in one letter, I think, in New Phytologist. The title is Concurrent Common Fungal Networks Formed by Different Guilds of Fungi. So it's broader than just mycorrhiza, and this is good. So the first question is, why do you think the focus on fungal networks has been so mycorrhizal center? Why the other kind of guilds have not been much studied regarding these networks?
And that's actually a question we've been asking ourselves, why that has really developed like that. I think there's no particularly good reason. I think there is a rather active research community of mycorrhizologists that is, interested in plant fungal interactions, and therefore, just by extension of that interest in what the mycorrhizal fungus does to the plant, there is also then this interest developing in what happens when you have more than one plant. And so I think this is why this happened. And I think is maybe more clear for root colonizing fungi with a positive effect. And there, your choices are not huge. I mean, the list is increasing clearly. But I think when you think about saprobic fungi, so decomposer fungi, I think most people do not immediately think of what happens if they link plants. And also the same is true for parasitic and pathogenic fungi. I think this people just don't think about it in this way. What is perhaps a little bit more surprising is that people working with endophytes also don't seem to be that interested in it, where, this is conceptually much, much closer to mycorrhizal fungi as the other groups. But yeah, in essence, that was the question we were asking ourselves. Why has there been this strong focus on mycorrhiza to the basically complete exclusion of all these other very interesting fungal groups? Yeah, that led us to write that second viewpoint paper that you just mentioned. Yeah.
And in the companion paper, in the other paper in functional ecology, I think it is, you make this distinction between common mycorrhizal networks with IFAL continuity and, I guess, just the broader category would be common mycorrhizal networks in general. Can you explain us these differences between both of them?
Yeah, so basically what we call the common mycorrhizal network with hyphal continuity, that is the current definition of a common mycorrhizal network used by other people. So the most broadly used definition. And when I and some other colleagues were thinking about this, we realized that it seems rather odd that we define a general phenomenon, namely the circumstance of mycorrhizal fungi connecting among different plants, that we define this more general occurrence basically by the specific case when there is, in fact, hyphal continuity from the root of one plant to the root of another plant. This is what's currently used by others for the definition of common mycorrhizal network. And so in order to give that the status that we think it deserves, namely the status of a special case, we propose this hierarchy of terms. And basically the lowest level of this hierarchy, so the most specialized case, is that hyphal continuity. And that simply pays tribute to the fact that mycorrhizal fungal hyphae can connect among adjacent plant roots in ways other than direct hyphal linkages with continuity maintained in the cytoplasm the entire time. There's also effects documented that when hyphae approach roots, there are already effects in that root. And there is for a bunch of the effects that we detail in that first opinion paper, no necessity for that hyphal continuity to be invoked in order for that connection to be meaningful. And so basically we have this broader category of common mycorrhizal networks. Above that is the common fungal network that we were just talking about, like why just limit this only to mycorrhiza, and at the highest degree of specialization and specification, we have this common mycorrhizal network with hyphal continuity. So that the idea is that when you really mean that, you are interested in that direct hyphal linkage with cytoplasmic continuity, then you just say that. But if you're more interested in other features of root systems being linked by mycorrhizal fungi or other fungi, then now we have also a term to be used. And maybe the last thing one could say is that we read that paper in lab meeting by Justine Karst and her co-authors, and of course it's an excellent paper, but in the end, it leaves you scratching your head like, oh my gosh, seems like almost impossible to do really work on this topic. And we had the feeling that may turn people off of this field of common mycorrhizal network. And we thought that would be kind of a shame. And so this was kind of a way to proactively get people on board to say, you can study these networks without necessarily having to invoke the hyphal continuity, which is extremely difficult to experimentally really show. I would say almost impossible, except for under very highly controlled circumstances, like in a petri dish or something. But you can't have it all. You cannot have mechanistic resolution to the highest degree, and you cannot at the same time also demand the ecological realism. That is fundamentally impossible. Nobody has ever achieved that for any topic ever. And so it's not possible to achieve it here either. So I think that has sort of precipitated that thought. But without that paper by Justine, we would have never thought about it. So I think it really precipitated that line of thought.
So two things that came to mind when you were speaking. The first one was that indeed for more than 25 years the focus and the current debate has been a lot in the IFAL continuity, and it makes me wonder. So which functional processes or which functions are manifested when there is functional eiffel continuity and when there is not eiffel continuity? Which do you think that functionally are the differences between these two?
Yeah, I think we'll list most of the things we could think about in this paper. I'm not sure I can like recapitulate them all, but I think when it is about carbon transfer from one root system to the other, I think then you really need to invoke, as far as I'm concerned, that hypercontinuity, because it is really carbon moving within, you know, getting jumping over to the hypha and then being moved along the hypha to the root system of that other plant, and then maybe subsidizing the mycorrhizal symbiosis in that other plant via the root system of this first plant. I think that is impossible without hypha continuity. And of course, it's a fascinating topic, and this is why people have been so interested in it. And maybe this has led to this becoming the actual definition of the broader phenomenon. And another example might be nutrients. Nutrients are being transported. I think that is also maybe difficult to conceive without that direct hypha continuity. And again, that is a topic where mycorrhizal researchers are very interested in looking at nutrient transfers. Even though I think if it leaves the hypha in between, at least in certain time scales, it could also be picked up by another hypha, and then, or if it gets dropped, so to speak, near the root system of the recipient plant, it might also get there in other ways in longer time scales. But you know that there's other processes where the hypha continuity is not needed whatsoever, and an example of that is, you know, transport of bacteria along the hyphae, or the transport of infochemicals that we've worked on along the hyphae, the transport of water along the hyphae, and basically anything that is transported along the hyphae. For all of these things, it doesn't matter if they're really connected directly physically from, if there's this continuity, you know, they can just move along close enough to that root system, and then an effect can be probably seen in the recipient root system. So I think this is some of the main processes that differ.
That's a great answer, and I think that of course our field is always very nutrient center, but of course there are many other functions and processes and signals and I don't know, ionosphere microbiomes that could be
Yes.
transported in the IV.
Yeah, exactly. I think that this is something that we're currently actually thinking about, is that from our systematic mapping exercise that Annika Lehmann headed, that revealed that, probably for the reasons that you just mentioned, very few people actually look what happens in the soil when you have a common mycorrhizal network established or not. And so I don't think we have a very good idea about the effects of the common mycorrhizal network on the soil. So if this is connected or not, what effect does that actually have on the soil? Because of the microbiome effects, because of soil structure effects, because of redistribution of pollutant effects and whatnot. So I think this is just currently not very well known. And yeah, you can look at that. I think is a very good question.
And the other thing that came to mind was regarding the experiments. So I got the same feeling from the article, from Justine's article and some subsequent articles. Um, the way I started to see this, the feel is that, yeah, you need, of course, the field studies, but they tell you part of the story, and then it's necessary to complement with greenhouse or lab experiments, which can tell you the other side of like the story. Um, yeah, it's always it will be always a big topic in ecology, and particularly in mycorrhizal ecology. Something that I also wonder is regarding the temporal aspect of the common fungal networks in general. So regarding because I imagine that the IV, I mean, I have seen some fine roots here in Chile that have really fast turnover rates, like 3 weeks, and then the IV, of course, are faster. So and this all I also always wonder about this in the first article by Justine. How long should the connection be, even if it's with IFAL continuity or without IFAL continuity, for there to be a functional effect or an effect in plant growth or in whatever?
Yeah, no, I think it's a great point. I mean, what we talked about in the paper basically as the little thought experiment in that first opinion paper was like, imagine, 1,000 typhal tips and they kind of hang out near that root but don't go in, and then one goes in, and then from that moment on you call that thing a common mycorrhizal network in the old definition, whereas before that you basically call it like nothing, because the connection hasn't been established. It doesn't seem reasonable. And so, but the point that you're making is equally valid. It's about the time, how long does a connection have to be in order for this to be effective and to be counted as this in this category. And it's just, it's just another example of how categories are, of course, very useful. And of course, common mycorrhizal network is a category where you have certain boxes that need to be checked in order to be counted as in. But categories are notoriously unproductive when things are very close to the borders of a category. And this is like one of these cases. So if there's a bunch of hyphae hanging out near that root system doing stuff, but they just don't go in, then that is still probably and most likely ecologically meaningful and interesting to study, but it's not inside of this category, hence it's not included. And I think that it is maybe an example of where categories sort of cease to be useful. And of course, we need always labels so we can say what we're talking about in any given paper. But when you sometimes these categories take on their own life, and then maybe it becomes also unproductive. This is also this change in nomenclature, and actually not just nomenclature, but also just concept, is an attempt to rectify that problem. Basically to say, well, there is still something happening, even if it's not connected. And the temporal dynamic you mentioned is just another excellent example that I don't think we have in our paper. Yeah.
That's great. And you just mentioned that, yeah, categories sometimes can be a box indeed, and can kind of dictate some research programs. In the new Phyto commentary or viewpoint you had, or letter, sorry, you also cited the work by Carlos Aguilar, saying that the lies between are blurred between different functional groups, specifically when talking about saprobic fungal networks and maybe symbiotic or mycorrhizal networks. What do you mean by that?
Yeah, I mean, that was a very nice paper headed by Carlos Aguilar Trigueros when he was still a student or PhD student or postdoc, I can't remember which it was, with us. And basically the idea was that a lot of these categories are in fact rather fluid. And, there's a saprobic fungus that basically decomposes organic matter in the soil, but maybe it can also go inside of that root, and maybe the root doesn't mind so much. If there's a little bit of a fungus inside of it, it doesn't do much damage. And so it will tolerate it. And most of the fungi that we isolate, they can probably move also inside of the root system a little bit. And then what does it become? And, one fungus can be maybe a parasite in one plant, but not a parasite at all in another plant. And maybe the parasite that is in one plant, it prevents a worse parasite from affecting that plant. What is then that parasite? Right. And I mean, there's many examples like that, where there's just basically rather a lot of fluidity in functional descriptions. And in that paper in Transient Plant Science from, I think, 2014, it was, we basically propagate and advertise the usefulness of a trait-based framework. So rather than calling this, a saprobic fungus, we say like, maybe its ability to have enzymes to decompose organic matter is high. Maybe its ability to grow in the root system is low, but it's not zero, but it can be quantified. And so once you dissolve these different species rather than putting them into rigid categories, once you dissolve these species into trait space, you can much more fairly deal with the true functional diversity that's out there. That's basically what this paper advertises. And that certainly has happened since that people are moving more towards a trait-based description.
That's very nice. And I think that, yeah, definitely we know uh, several phenomena in mycorrhiza and in saprophytic fungi, which indeed these differences can be, there is not like a straight line in some cases, in some special cases. Um, to finish up, two questions. Can you elaborate a little bit? Um, we have been talking mostly about mycorrhiza, so can you elaborate a little bit on parasitic fungal networks and on saprobic fungal networks?
Mm. Yeah, so I think that there is not much out there. It could be a very short discussion, but I mean, there are papers that show that parasitic fungi, when they infect plants, when the infection emanates from one root system, moves to the soil and infecting other root systems, that it is very likely that that fungus is still also in that other system. And so that maybe they really also have this continuity of hyphal presence in the soil, maybe even. So that there is linkages among fungi, among roots also by parasitic fungi. Has not been studied, to my knowledge, explicitly. I mean, we may have overlooked some papers, but papers that I found that basically is when there is an infection emanating from a root system. This is basically epidemiology of an infection that has been looked at, quite a few papers actually. And it seems unreasonable that that fungus basically is no longer there once it infects the next plant. So implicitly in these studies, even though they don't really mention it and don't examine it, it seems like there could be links made by parasitic fungi, which are also root colonizing fungi. The same is probably true of endophytic fungi. And for saprobic fungi, I think this topic has just not been really touched because it is a bit counterintuitive, and I think hesitated a bit putting it in the paper, I think. But when you think about it, they're also probably taking carbon from the rhizosphere and they're linking to the root system of a plant nearby. And thereby they could be transporting things, and that connection could be meaningful. And by their ability of having tapped into two sources of carbon for them, they may have also affected the soil in between them and the microbiome that they carry and influence. So it seems not totally impossible that they have some sort of effect in linking these root systems. To me, it seems plausible. But I think that is, not quite so easy to study potentially.
And that brings me to my last question regarding the importance of the process or the framework of syntropic interaction. So it seems like what you just described is like a concatenate effect, and maybe that is a useful framework for mycorrhizal ecology in general. We have been focused uh, in two actors, but it seems like we need to invoke and include many more actors in these interactions.
Yeah, I mean, I think we give some clues to that in the paper that maybe a mycorrhizal fungus is much more effective in that linkage if it, so to speak, teams up also with a saprobic fungus that maybe mineralizes some nutrients that then can be transported, or that entire microbiome interaction as it plays out as a whole, co-metabolism, as you mentioned, but also various interactions among these functional groups. I think you can think of many, mycorrhizal fungi and parasitic fungi. This has been studied quite a bit. How mycorrhizal fungi protect the plant root from parasitic fungi. What happens when this happens in the context of common mycorrhizal network in the wider sense, in the broader sense? Can a connected mycorrhizal mycelium maybe more effectively protect the plant against pathogens. Is a pathogenic network more powerful because it's hooked into multiple plants? Those are some questions one could ask. And how do these various networks interact with each other? I mean, the picture that emerges in my mind is really that, of course, it's commonly accepted scientific practice to break down a complicated system into its component parts and then study them. But then I think in the end, it is at your own peril if you don't make some effort to put it back together again. And it's entirely possible that these various common fungal networks done by various groups of fungi, functional groups of fungi, they could interact in really meaningful ways.
Mm. Sure, I completely agree with this last conclusion that for studying things we break down, but then it's immediately necessary to to put them back together, all these components. So thank you very much, Matthias. This was a very interesting interview, and thank you all for listening. And see you next.
Thanks very much.